Good evening, everyone. Emory Moynagh here for Creation Biology, and it's been about a week since we were able to do a recording, and the reason being for that is I've been, fairly sick, and I'm just now getting my voice back, and as you may have noticed, that the voice is significantly lower than the previous tone, than my previous normal voice. Hopefully that won't be too much of an annoyance, because we're going to read Walter ReMine's Discontinuity Systematics paper today. This is from 1990, so a significant paper from the early days of baraminology. Now, I do want to be fair. I am going to read Kurt Wise's paper as well from 1990. There were the two major baraminology papers in 1990. Which I'm going to read at some point. I just decided to start with ReMine, in part because I said I was going to. ReMine's paper is about 10 pages long. That includes references, so it shouldn't take us too long to get through all that. But I will be interspersing commentary as well. Also, I'm planning on reading Galuza and Gaskell's CET model paper. That's 28 pages, so that'll probably be a James Lindsay-worthy podcast. This one, however, should be a little bit shorter. All right, so let's go ahead and jump into the fun stuff. Starting with the paper, the title of the paper, which was submitted to the International Conference on Creationism in 1990, Discontinuity Systematics, a new methodology of biosystematics relevant to the creation model. Abstract. According to the creation model, an important feature of life is discontinuity, the discontinuity between the originally created life forms. Yet all existing methods of biosystematics are inherently incapable of recognizing or even describing the discontinuities of life. To meet these needs, a new method of biosystematics is proposed called Discontinuity Systematics. 4 new terms are introduced: holobaromin, monobaromin, polybaromin, and apobaromin. These terms allow for the identification, description, and evaluation necessary in the new systematics. The special interrelationship of the terms allows biosystematic knowledge to be constructed in a methodical way. Lineage, reproductive viability, biological experimentation, and similarity are discussed, showing how they assist in the identification of groups in the new systematics. Discontinuity systematics will aid discussion of a significant biological system pattern and begin the accumulation of evidence relevant to the creation model. All right. So there's a lot in that abstract, and we will go through a lot of this stuff as we go through. But just to point out, ReMine, some of those terms may sound familiar to you if you're familiar with baraminology. Wise borrowed some of the terms: holobaromin, monobaromin, apobaromin. I don't recall if he borrowed polybaromin. We'll look at that when we do Wise's paper. But some of the terms did come from ReMine. And the importance of discontinuity also comes from ReMine. And just in case you don't know what discontinuity is, it means it just means difference. It just means that there's not a continuous line from one species to the next. That's what it means, roughly speaking. I'm not going to go into the whole technical details here. Introduction. The difference between the modern views of creationists and evolutionists is discontinuity, and that is true to an extent. So I'm leaving Walter ReMine here, and we're going to opine for a moment. The creationist model does require significant discontinuity. A lot of discontinuity needs to be present in the creationist model. However, the evolutionists do themselves argue for some limited amount of discontinuity. So it's not just the existence of discontinuity, because if there was no discontinuity, then every species would grade directly into each other. You wouldn't be able to tell any species apart. So the evolutionist model does require some discontinuity, however, not nearly as much as the creation model. So perhaps the better way to phrase that would be that the creation model requires more discontinuity than the evolutionary model. In the creationist view, many groups of organisms are separate and distinct, disconnected from other groups. In contrast to the predictions of evolutionary theory, creationists feel that life displays an important pattern of discontinuity. Yet currently there is no method of biosystematics which is capable of identifying and studying discontinuity, and that's the problem ReMine wants to solve. All traditional methods of biosystematics are insensitive to discontinuity and inherently incapable of identifying it. They are, as it were, blind, quote unquote, to discontinuity. Phylogenetic systematics and evolutionary taxonomy explicitly assume continuity and thus always conclude that continuity is a characteristic of life. The methods of phonetics and transformed cladistics produce data structures such as phenograms and cladograms. These data structures locate life forms at the tips of branches of a tree-like diagram. Many people erroneously identify such diagrams as with an evolutionary tree, phylogeny, and thus prematurely presume the evolutionary continuity of life. Nonetheless, these systematic methods do not try to determine whether or not discontinuity actually exists. So it's important to note here, when we eventually read the Robinson and Kavanaugh paper from 1998, where the barometric distance was introduced, they admit freely to relying heavily on phonetics. ReMine doesn't seem to want to do that. ReMine wants to avoid phonetics. He seems to want to... there seems to be a lot of different things that ReMine wants to do a little bit differently. We'll get more into that as we move forward into the paper. A biosystematic method for identifying discontinuity would help scientists study this important aspect of life's pattern. This would also enable creationists and evolutionists to more clearly communicate information and viewpoints. This paper proposes such a method for the study and description of the Earth's biota. The method is called Discontinuity Systematics because it focuses on the discontinuity as a pattern of life. Discontinuity Systematics seeks to identify the boundaries of common descent. So, this is a limited common descent model. The idea is that there is some common descent, but the descent is limited. And so they're trying to find the boundaries of that descent. It is similar to the baromenology model that, of course, became popular because of Kurt Wise. Walter ReMine had several things working against him. First of all, Kurt Wise was a Harvard PhD. He studied under Stephen Gould, so he had a lot going for him there. Second, ReMine was more closely aligned with the Discovery Institute and the ID movement than he was the creationist movement. Third, he was only had a master's degree, and so everybody ran with Wise's ideas and not ReMine's. Even though Wise did borrow from ReMine, not inextensively, but not entirely. I mean, Wise does have his own ideas, and we'll get to that when we read Wise's paper. But Wise does borrow some from ReMine. New header, kinds. Many creationists have used the term kind for their biosystematic unit. However, this is an inadequate term for discontinuity systematics. Many anti-creationists have cogently argued that kind is an ill-defined and ambiguous term. They still make that argument. I would argue that the term is not as ill-defined as it could be, but even if it is ill-defined, we could just flip it around and say, okay, well, define a species. Tell me what a species is. Last count I had, I think there was 36 different definitions of species. It's so bad that there's one species, one guy who studies species and speciation, who actually made the quip, and I can pull the quote up at some point, who said something along the lines of, in a room of N biologists, there's N plus 1 definitions of species, because nobody knows what a species is. So if they want to argue that, a kind is undefined or biologically irrelevant, well, okay, a species is undefined and biologically irrelevant. Boom, done. Moving on to ReMine. First, confusion arises from the history of the words, species and kind. At one time, the two words were synonymous. In fact, species was merely the Latin word for kind, and that's true, by the way. Before Darwin, some scientists used the term species with virtually the same meaning that creationists use the kind today. For example, von Baer in 1828 defined a species as the sum of individuals that are united by common descent. Even today, some people equate kind with species. However, the term species has been redefined in many new ways by modern scientists. There is now a wide variety of species definitions, and significant disagreement exists about which of those definitions are most appropriate. Well, that's true. and by the way, even the Evos will back me up on this. I appeared on Dr. Dan's channel for one particular episode of his discussion. We had a discussion with creationists. I think it was an AMA episode. And I actually asked him about the species thing, and he kind of laughed, and he said, if you want to have something along the lines of, if you want to have a fight in a journal club, make it about the definition of a species. And he's right, by the way, because it's an absolute joke. Nobody knows what a species is. The modern term species carries much unwanted baggage of semantic ambiguity and confusion. Therefore, equating kind with species only results in further confusion. Now, here I agree. I don't think that kind and species are equivalent. I don't think that they should be equivalent, and I don't understand why some people continually think that they are equivalent. 5 seconds of reading in the young Earth creationist literature would explain this. Now, this may not have been the case in 1990. That was 33 years ago. Holy jumping. That was 33 years ago, but it is highly relevant today, that the fact that we're talking about species, and still talking about the difference between species and kind. Second, there are several colloquial non-biosystematic definitions of kind which lead to ambiguity. Third, kind has several conflicting biosystematic definitions, once again leading to ambiguity. Fourth, a single term like kind is insufficient for doing serious work in biosystematics. Fifth, several interrelated terms are necessary for precisely conveying the results of biosystematic research. Now here, I don't necessarily disagree with him. Different people have defined kind different ways. I do think there is probably a good way you could get kind out of Scripture, well, at least a decent definition, but I don't see that there's a problem with his choice of terminology. All right, new header, new terminology. Frank Marsh, 1947, by the way, round of applause, citing Frank Marsh. Well done. Of course, this was in 1990. Marsh was still alive. Coined the term baramin to mean created kind. Marsh constructed the term as a compound of two Hebrew words, bara meaning created and min meaning kinds. This unique word serves as a root for the new terminology of discontinuity systematics. Discontinuity systematics classifies only real known organisms, not hypothetical, imaginary, or undiscovered organisms. Discontinuity systematics classifies known organisms into groups. These groups are defined relative to all known life forms, fossil or living. Each group includes only known life forms and excludes only known life forms. Thus, these groups can neither include or exclude organisms that remain undiscovered. Therefore, as new organisms are discovered, some specific groupings would need to be adjusted accordingly. For example, if a certain group is said to contain all its ancestors, then this refers only to known data. As new ancestors are discovered, they would need to be added to the group. And this makes sense. You can't classify something that you don't have data on. And if we haven't discovered the organism yet, then we don't have any data. So you can't make any classifications based on that. There are 4 types of groups defined as follows. And here's the first definition. Holobaromen. A complete set of organisms related by common descent. A group containing all and only those organisms related by common descent. This may be taken to represent a set of organisms directly originated as a single reproductive unit together with all their descendants. Okay? So this is your original created kind, your original reproductive grouping. That's a holobaromen. Now, I honestly do not... I have a hard time using so many words for this. I don't think it needs to be quite this complicated, but I understand why ReMine is doing what he's doing here, and there is a place for complicated terminology. I don't think abandoning the word baramin completely is necessary, but I understand what he's trying to do. So a holobaromen, I honestly think he could have just called this a baromen and been done with it. But this particular one anyway. A holobaromen is effectively your created unit. A monobaromen, which is the next word, a monobaromen containing only organisms related by common descent, but not necessarily all of them, i.e. a group containing an entire holobaromen or a portion thereof. So in other words, this would be if you take the canid lineage as including the foxes and the dogs, then the foxes would be a holobaromen and the dogs would be a monobaromen. Sorry, they'd be a monobaromen. So the foxes would be monobaraminic and the dogs would be monobaraminic because they both would be related by common descent. Each group internally is related by common descent, but they don't make up an entire baromen, so they're not a holobaromen. Hopefully that makes sense. So for an apobaromen, it's a group of organisms which contains all the ancestors and descendants of any of its members, but which may contain subgroupings that are unrelated to each other. So this is a little bit interesting. I'm not entirely sure why this particular group is needed, because effectively this is a group containing one or more holobaromens. But if a holobaromen is the original created kind, which is usually the synonym we go with, then I'm not entirely sure what good an apobaromen is when we talk about a polybaromen as our next one. And the definition of a polybaromen is a group of organisms which does not share a common ancestor, i.e. cats and dogs. So you'd have multiple holobaromens. I'm not entirely sure. I think apobaromen must be a fairly specialized case, because I'm not entirely sure how usul that would be. But hopefully ReMine will explain that as we go further along. Remember that these are specialized terms, defined for use within discontinuity systematics. They are defined as referring only to known organisms, that is, they include and exclude only known organisms. Again, he's being a little bit repetitive here. I don't think he needed to repeat that. But at any rate, this was again 1990, so things were different then. Each of these above four terms was followed immediately by its definition. These definitions are used by the discontinuity systematists to identify groups of organisms. Along with each definition, a secondary meaning is provided within parentheses as a comment. The secondary meaning shows how the terms are interconnected and ultimately how they may relate to creation theory. The special interconnection of the terms allows biosystematic knowledge of life's pattern to be constructed in a methodical way. So again, this is just building a foundation here. This is building up to what's coming on. Right? The plural form of each term is constructed by adding an s, e.g., holobaramins. The adjective form is constructed by adding an ic, e.g., mammals are apobaraminic, dogs and wolves are monobaraminic. When a group is identified, it can be communicated to other researchers as a list of recognized organisms, or the group can be given a specific name according to the organisms it contains, just as current taxa are named according to contain subtaxa. For example, the placental dogs, coyotes, wolves, foxes, and jackals can be called the canid monobaramin. If existing formal taxonomic names, e.g. Canidae, are used, then they should be used cautiously. This is because existing formal taxonomic names have been defined by means of another biosystematic method and can vary somewhat arbitrarily as the biosystematic method develops. So this I agree with. What ReMine is saying here effectively is, hey, if we're going to use the existing systematics, the existing names for organisms, group names that is, we need to be careful with that because the group names may not be accurate because they've been used with a different systematic method. And given that evolutionary trees and cladistic classification has been going on for the last close to 100 years probably at this point, it is quite possible that many of the groups are incorrect. I know Todd Elder likes to talk about how the plant groups are absolute junk because they've been used with, they've used so much cladistics and phylogenetic trees to build their groups. So just something to be aware of. And now I don't see that this was ported over. Most baraminologists gladly use group names. I don't see that this was ported over, and it's something that needs probably to be addressed. All right. The modifiers can be used with the terminology to more precisely identify groups of interest, e.g. the Devonian shark apobaramin, the living canid monobaramin. This basic terminology is sufficiently versatile to identify, describe, revise, and discuss taxonomic groups within discontinuity systematics. Discontinuities should interest evolutionists since these might represent punctuational or saltational events of rapid undocumented evolution. Again, he's writing in 1990 when Stephen Jay Gould is still alive, still teaching at Harvard, and still putting forward his ideas about saltation. Those have kind of gone out of favor now that Gould is no longer alive to promote them. Which is kind of unfortunate because I did like kind of having the evolutionists at each other's throats. Moving on. Discontinuities would require special attention and explanation from evolutionists, and the precise identification of discontinuities would be the first step in their understanding. Thus, the new systematics could interest those evolutionists who boldly seek to test their theory or understand it further. There aren't very many of those. That's editorial. Moreover, discontinuity systematics has a clear impact on the origins debate. Evolutionists claim that all life arose from one common ancestor. Thus, they would claim that ultimately there is only one holobaramon. Modern creationists claim that numerous life forms were separately created, varying somewhat thereafter. This suggests that we should find many separate holobaramons. Therefore, the discovery of a clear and consistent pattern of numerous separate holobaramons would be a major evidence for creation and against evolution. Not that they'll ever admit it. The new biosystematic terminology interfaces with creation theory in an uncomplicated way, as discussed next. Baramin is a term sometimes used in creation theory, meaning a group of organisms directly created as a reproductive unit and all their descendants. Creation theorists use this term in their theories to explain 1. The originally created pattern of life, and 2. How that pattern has varied since creation. Now, I think this has changed a little bit, because Baramin has, in the research world, Baramin has kind of faded out to a certain extent. Holobaramin tends to be used more frequently. Baramin is not used as much anymore. Um, but it is still used in common parlance quite a bit. So it is useful to still remember the word, but it's no longer used as much in the research terminology. So, moving on. Yet baramins have been difficult to study scientifically. One difficulty has been identifying and delineating the baramins. Another difficulty is that most individual organisms are undiscovered. They either perished without a trace, or their traces have yet to be found. Thus, most organisms from a baramin remain undiscovered and cannot be known. These factors have hampered scientific study of baramins. I'm not entirely sure that most living organisms perished without a trace. I actually think we have a fairly relatively complete fossil record at this point. We've got fossils of most organisms that have probably lived. Now, I'm sure we're missing some, particularly soft-bodied organisms. But, I think our fossil... Now, of course, he was writing 30 years ago, so that things have changed somewhat. I'm not entirely sure I agree with this. I think that at least that particular objection. Now, I do agree that it has been difficult to identify and delineate baramins. That has been difficult, and it's been done very poorly for the last 30 years. But roughly about the same time that ReMine wrote this paper, but it's not ReMine's fault. That said, I don't think that the other objection is necessarily valid. I do think that the fossil record is in a fairly decent state at this point, and that, we have a fairly good grasp on the organisms that once lived. I'm sure we're missing some things, I'm sure we'll find more things, but I think we have a fairly good grasp on the organisms that once lived. However, a holobaramin is comprised solely of known organisms, and therefore it lends itself to scientific investigation. Moreover, a relationship between holobaramin and baramin tentatively suggests itself. A holobaramin tentatively may be taken to represent those members of a baramin who have been discovered. Thus, the term holobaramin from an empirical science of biosystematics has a suggested connection with the term baramin from creation theory. And this is important to understand. This is kind of why the term baramin has faded out in the literature to a certain extent. Because holobaramin and baramin are roughly equivalent terms. Effectively, pretty close. All right, let's get into the methodology of this theory that ReMine was putting forward. Discontinuity Systematics seeks to collect organisms into identifiable groups. It seeks to eliminate all polybaraminic groups and instead identify groups that are monobaraminic, apobaraminic, or better still, holobaraminic. Our knowledge of monobaramins is improved by additively combining them together into a larger monobaramin. On the other hand, our knowledge of apobaramins is improved by subdividing them into smaller apobaramins. The ultimate goal of discontinuity systematics is the identification and description of all holobaramins. Holobaramins are identified through a process of successive refinement. Since every monobaramin is a subset of a holobaramin is approached as a monobaramin is successively increased in membership. For example, as more members are added to the canid monobaramin, and he gives examples, the placental dogs, coyotes, foxes, wolves, and jackals, the holobaramin in which they are found is gradually approached. On the other hand, since every holobaramin is a subset of an apobaramin, the holobaramin is also approached as an apobaramin is subdivided into smaller apobaramins. For example, the mammalian apobaramin is successively subdivided into smaller apobaramins. The holobaramin containing placental dogs is approached. In this way, the successive increase of the placental dog monobaramin and the successive subdivision of the mammal apobaramin will converge on the holobaramin that includes dogs. Thus, holobaramins are identified by successively refining our knowledge of monobaramins and apobaramins. So effectively, this is top-down and bottom-up. You start with a few species in a monobaramin that you know are united by ancestry, and then you say, okay, can we combine those with any other species that might be united by ancestry? Oh, these other things are, so we can increase the size of our monobaramin. And then we just increase it upwards until you get to a point where you've decreased the apobaramin down, and you increase the monobaramin up to a point where you have a holobaramin. Now, membership criterion. This is where it's going to get interesting and important. An important aspect of discontinuity systematics is the membership criteria. The criteria that determine when an organism is or is not a member of a certain group. Group membership is based on continuity through common descent. Therefore, the membership criteria seek to identify continuity and its boundaries. Continuity and discontinuity are related observations. You cannot see one without having eyes to see the other. Thus, to see discontinuity, you must have a way to see continuity, and the membership criteria supply this capability. The membership criteria are intended as a consistent set of tools for recognizing continuity and discontinuity. Discontinuity systematics seeks to identify the boundaries between common descent and discontinuity. It views all available scientific evidences as legitimate with two explicit clarifications. First, discontinuity systematics is independent of creation theories, that is, the methodology uses no outside knowledge of the biotic pattern originally created or intended by life's designer. Rather, the methodology attempts to discern life's pattern as seen through a neutral scientific observer. Now, here is where ReMine's tie in with the DI and intelligent design comes front and center. I strongly disagree with this, because I strongly disagree that there is a completely neutral observer. Even the evolutionists don't believe this. Now, I think that you can approach some form of neutrality, and you can recognize your bias and work against your bias, but take steps to guard against your bias, but a purely neutral observer doesn't exist. Everybody's got some kind of bias somewhere around. ReMine here, I think, is trying to build something that is completely independent of creation, and I got news for him: because it was written by a creationist, the evolutionists are still not going to accept it. It's just that simple. They're not going to accept it. It doesn't matter. So you may as well just be openly creationist. So I think that's a underlying potential problem with discontinuity systematics. Potentially. Second, discontinuity systematics holds that cladograms and phenograms are inconclusive as evidences of evolutionary continuity. These diagrams have the appearance of an evolutionary tree of phylogeny, yet they are not. In fact, they fail to identify any real ancestor-descendant relationships in the data. The specification that cladograms and phenograms are inconclusive merely formalizes a view already held widely by many scientists. This specification can be further justified. Several adequate methods already exist for studying the phenogram and cladogram patterns in nature. Discontinuity systematics acknowledges the existence of these patterns and formally sets them aside so that the remaining pattern may be clearly examined. Thus, discontinuity systematics studies a pattern that is unstudied by any other existing systematic method. Now here, I think ReMine is giving a little too much credit to the evolutionary model. Now, I know ReMine knew the model pretty well, and there's actually a population genetics paper that he wrote in the Journal of Creation. I don't think it was the Journal. It might have been the Journal of Creation at that point. I don't remember now. That I want to read at some point. It's going to be hard. It's going to be a slog. And probably not a lot of people will listen to that particular episode. But I do want to read that paper because I think it's very interesting. But ReMine is giving too much credit here to phylogenetics. Phylogenetics is a useful tool to a certain extent, but if you want to promote a particular idea in phylogenetics, you can. It's real easy. It's real easy to make the data say whatever you want it to say. I could do it probably in 20 minutes if I worked at it hard enough, maybe a little bit longer, to make things that are definitely not related. So he's being a little bit too generous, I think, to the evolutionary model here. But that said, we can grant that because this again was 30 years ago. In short, the methodology tries to identify the boundaries of evolutionary continuity by emulating a neutral scientist who, 1, has no detailed knowledge of creation theory, and 2, views phenograms or cladograms skeptically or agnostically. Discontinuity systematics has a strong bearing on the evidence for creation and evolution, yet it gives special privilege to neither theory. Again, the evolutionists aren't going to like you for this, bud. And I'm sorry, but they aren't. It doesn't matter. You can't play this neutrality game. You may as well be all in. Discontinuity systematics is a methodology for neutrally examining an important pattern of nature and communicating the results. This new biosystematics is an empirical science that uses only data observable with the senses. Groups are identified in a tentative manner, and these may be challenged, debated, and revised based on the available data. The membership criterion provide the means by which groups are tentatively identified. The membership criterion are crucially important to discontinuity systematics. A few of these criteria are discussed below. And now we're about to get into a list of the the things that ReMine considers to be important. The things that ReMine considers to be important in determining relationships in his discontinuity systematic model. Now, a lot of these things have not been ported over. So, the modern baraminology uses basically none of these, or very few of them. Okay? So let's get into them. First one is the lineage criterion. Virtually all lineages, and he puts that in quotes, and phylogenies, he puts that in quotes too, offered by evolutionists are not lineages, rather they are cladograms and phenograms having an appearance, falsely, of an evolutionary tree. These fail to identify real ancestor-descendant relationships in the data, therefore they are not lineages. So in other words, what he's saying is that most of the evolutionary trees that you see in your textbooks are a joke. They don't actually show you anything about ancestry, they just show you a cladogram and say that this is the evolutionary classification of these things. Organisms may be viewed as data points within a multidimensional morphology space. Oh boy, okay, this is where I'm gonna have issues here, but we'll continue. Lineages must curve their way through a morphology space with ancestors and descendants in succession. A nondescript cloud of data points in morphology space is not a lineage. Rather, a lineage must have a special pattern. Oh, okay. This is interesting. Let's finish this. Rather, a lineage must have a special pattern. A lineage must be a trail of data points, long and narrow, with an absence of data points in region adjacent to the lineage. If two organisms are connected by a clear-cut lineage in morphology space, then this qualifies as sound empirical evidence that they are in the same monoboramen. If a lineage is sufficiently clear-cut, then it can unite organisms into a monoboramen, even if there are large morphological distances between the data points in the record of life. This criterion only requires that the data have a special type of pattern, a lineage. This criterion is quite powerful and in principle could span large, quote-unquote, gaps in the record of life. Pause. That paragraph right there. So, there is some talk within the statistical baraminology community of 3D morphospace. However, they talk about characters in 3D morphospace, not taxa. And taxa, they talk more about characters in 3D morphospace than they do taxa. And there's another paper that I need to read at some point. The redefinition of what a baramin is from Todd Wood, Kurt Wise, Neil Duran, and I forget who the fourth author was. But I think it might have been Roger Sanders. We need to read that paper at some point too. But this idea of a line, a lineage of being able to track through time or track through morphospace, that's really interesting. However, ReMine goes on in the next paragraph. I'd like to see a program capable of doing this, just to see if it would work. So if anybody out there is a programmer and wants to write this, here's an option for you. Nonetheless, I suggest that life generally fails to be joined together by clear-cut lineages. Lineages do not span life on a large scale. In most cases, evolutionists cannot even agree among themselves about the ancestors of a given group. Moreover, life lacks clear-cut lineages, especially at those places where they are the most desired by evolutionists, at the origin of new major biotic designs. I suggest that large-scale lineages are systematically lacking from the record of life. Discontinuity Systematics seeks to precisely document the situation by identifying the boundaries of continuity. This is one of the major challenges of the new biosystematic. Now, what he seems to be saying here, and I could be reading this wrong, but what he seems to be saying in this paragraph is that the massive tree of life lineages don't exist. However, I don't see that precludes within baramin lines, within holoboramen or monoboramen lines. I'd like to see, if someone, maybe if Dr. Matthew Trahady sees this might be something that would be worth playing around with. The idea of a line of organisms in ancestry, a lineage that shows up in morphospace. Because right now what we've got is the 3D MDS plot, which is rotatable. You can rotate that however you want. This is something that I might be able to do something with. I got to think about this. There's a lot here And I think that this idea was never ported over into statistical baraminology, and it probably should have been. I think it would have been interesting to see if it worked. I don't know if it would work. I'm not a programmer. I couldn't program this to save my life. But I think there's something here that borrowing from ReMine here might be worth testing, if nothing else. It may not work. I don't know if it'll work. But I'd like to at least see someone test it, which I don't think anyone's ever done. Statistical baraminology is not good about testing new things. They just kind of throw things at the wall and see what sticks. All right, next criterion, reproductive viability. Reproductive viability is the ability of two organisms to interbreed. Reproductive viability is a membership criterion that is already widely used by creationists. Not anymore. This criterion also plays a crucial role in the definition of most traditional biosystematic units. Again, not anymore. Well, I guess for traditional biosystematic units like a species, yes, it would. Reproductive viability is often complete, yielding viable, fertile offspring. Such a circumstance is sound evidence that the two organisms are of the same monoboramen. Although this criterion is a good one for identifying monoboramens, there are some difficulties to be discussed. Reproductive viability is sometimes incomplete or partial. For example, hybrid offspring may be viable but infertile. This occurs when horses and donkeys mate to yield a mule. The mule is a healthy, viable organism, but it's sterile. That's not entirely true. There are a few cases where mules are fertile. Not very many, but they do exist. Nonetheless, most creationists feel that this case has sufficient evidence to place the horse, the donkey, and the mule in the same monoboramen. And yeah, I would agree. That's pretty straightforward and simple. If they if they're able to breed and produce even sterile offspring, they're of the same monoboramen. In ReMine speak. Partial reproductive viability is sometimes difficult to assess. For example, when a mating between two species yields inviable offspring that do not survive even to birth. These cases need more research. There has been a little bit more research in these areas. Gene Leitner has done some. Sigrid Schur has done some as well. Overall, I think we're looking at like a... Oh, good heavens. Leitner's criterion was further on than Schur's. Was something along the lines of a certain number of cell divisions. It may have been 8 cell divisions. I don't remember exactly. That's another paper I need to read now. But a certain number of cell divisions are qualified as a creative kind. Or holoboramen or monoboramen, I guess, in ReMine speak. Even more difficult to assess are cases where man has artificially forced the genome of one species into another species to form a hybrid. For example, man has used gene recombination techniques to place human genes into bacteria. Likewise, viruses can sometimes transfer small pieces of genetic material from one species to another, into quite different species. Such hybridization is very fragmentary and partial, for it's the mixture of minor parts of genomes from different organisms. Presently, most creationists feel that such severely fragmentary and artificial hybridization fails to unite two species into a monoboramen. I would agree. I don't think that's any kind of... honestly don't even think that needed to be brought up. I mean, I understand why he did. He's trying to be complete, but I don't think that needs to be brought up at all. It's pretty obvious that gene transfer is not the same as reproduction. Gene transfer is not the same thing as reproduction. You actually need to have some form of reproduction success beyond fertilization for reproductive viability to be achieved. Thus, reproductive viability spans a range of outcomes from complete to fragmentary to incomplete. More research must be done to further develop this membership criterion. I agree with that. There needs to be more research on that. However, since statistical baraminology has completely abandoned the reproductive criterion, it probably won't be. Using reproductive viability as a criterion, the horses, the mules, zebras, and onagers are united into a monoboramen. Lions and tigers are placed into a monoboramen, as are cattle, buffalo, yaks, and bison. Mallards and pin-tailed ducks are united into their own monoboramen, as are placental dogs, wolves, coyotes, jackals, and foxes united into their own. One of the first tasks for discontinuity systematics should be the documentation of all such monoboramens. So in other words, the documentation of all reproductive groups that we know of. And I think this has been done to a certain extent in the Archekinds articles that Answers in Genesis did. Anytime there was hybrid data that was available, they used it. As such, I think that this may have been done. This particular thing may be complete. Overall similarity. Presently, the measurement of overall similarity seems to be an interesting, though often unreliable criterion for determining common descent. Even at the level of DNA, measurements of overall similarity can give results that are difficult to interpret. For example, there are two Drosophila species that are morphologically quite similar, yet the DNA of these two species are 30 times more dissimilar than the DNA differences between chimpanzee and human, which are morphologically more distinct. Thus, overall similarity of DNA does not necessarily correlate well with overall morphological similarity. That's true, by the way. Moreover, there does not appear to be a clear threshold of overall DNA similarity that would consistently indicate the presence or lack of common descent. The measurement of DNA similarity is yet in its infancy, and its role at this time is unclear. Again, this was 1990. Okay, so DNA research has been done, quite a bit of it since then, and ReMine's comments on this are indicative of he was writing this in 1990, probably wrote it in 1989, or at least part of it. Several types of overall similarity measurements might be helpful as membership criterion, but much research needs to be done to determine how to use them. There has been some work done in this area. Roger Sanders put together oh, good heavens. Well, it's a criterion that basically uses the similarity of the organism as a proxy for relatedness. So that has been done. I can't remember the name of it right off the top of my head, but that has been done to a certain extent at this point. So that's another paper we might read at some point. The experimentation criterion. Experimentation is strong evidence for demonstrating the likelihood of common descent. When a breeding experiment produces a range of new morphologies, then this range becomes a standard by which we can measure the morphological differences between comparable organisms. For example, if interbreeding two organisms creates a diversity of morphology which reasonably overlaps that of a third organism, then there is reasonable evidence that the third organism belongs in the monobaromen of the first two. By analogy, this criterion can be cautiously applied to fossil organisms. Suppose two living organisms are comparable to two fossil organisms. If interbreeding of the two living organisms produce a range of morphology greater than the difference between the two fossil organisms, then there is evidence that the two fossil organisms belong together in a monobaromen. If the members of a monobaromen define a region of multidimensional morphological space within which a test organism falls, then there is evidence that the test organism should be included in that monobaromen. Other membership criteria will undoubtedly be developed, and these will be an active area of discussion and research. Creation should pursue the identification and refinement of such criteria. This has not been done at all, unfortunately. Basically, no one has touched this because ReMine's work is sitting on a shelf somewhere gathering dust, and that's unfortunate because ReMine is probably one of the best thinkers in systematics that creationists have ever produced, and we've effectively ignored him. The identification of apobaromens. So far, this paper has discussed the criterion used for joining organisms together into one evolutionary unified group, a monobaromen. This same consistent set of criteria are also used to identify an apobaromen. In particular, an apobaromen is identified because it fails all membership criteria that would connect it with any other group. An apobaromen is a separate, distinct group that is unrelated to any other group. If a group fails to demonstrate reproductive viability with any non-member, and if there is no clear-cut lineage linking the group with non-members, and if biological experimentation fails to span the gap between the group and non-members, then there is sound empirical evidence that the group is an apobaromen. In short, an apobaromen is a group which empirically fails to be evolutionarily united with any known organism outside the group. I suggest that life is comprised of numerous apobaromens. A discontinuity systematics seeks to study this situation and communicate the results. This matter should be of interest to all scientists concerned with the origins debate. Now, the point here that he's making effectively is that certain groups obviously don't lump with certain groups. Like dogs and cats clearly are different from each other. However, they're similar enough that they could both be considered carnivores. They're, order Carnivora. So order Carnivora would probably be considered an apobaromen. Because everything within the group is somewhat similar. But everything outside the group is, well, very different. So that's probably the best way to think about an apobaromen. Again, I don't think apobaromen is completely and 100% necessary. But hey, this was what ReMine was thinking in 1990. Okay, so things have changed since then. That gets us to the conclusion. Discontinuity systematics provides the only systematic method for identifying and studying the discontinuities of life. Discontinuity systematics is a methodology for studying this pattern from a neutral point of view. Yet as this pattern is systematically documented, it can provide substantial evidence for the creation model. In addition, the new biosystematics provides creationists and evolutionists with the terminology necessary for convenient discussion of their viewpoints. Discontinuity systematics produces only 4 new terms. The interrelationship among these terms allows for the knowledge within the field to be built in a methodical way. Holobrahmins are identified by successive refinement through the convergence of monobrahmins by addition, and apobrahmins by subtraction. As more researchers study discontinuity systematics, the membership criterion will be improved and overall subjectivity will decrease. The terminology of discontinuity systematics is versatile enough to allow for that kind of perpetual improvement. Discontinuity systematics stimulates several types of research. One important research project would be the identification of monobrahmin groups based on the criterion of reproductive viability. Another research project could evaluate partial reproductive viability as a criterion for identifying monobrahmins. Yet another research project could begin a preliminary identification of apobrahmins by looking at higher taxonomic levels and recognizing the largest and most certain discontinuities. For example, whales and bats each seem to be a coarse yet defensible apobrahmin. Another project could review comparative DNA studies and evaluate their significance to discontinuity systematics. Can overall DNA similarity be systematically used to identify monobrahmins and or apobrahmins? Another project would be the evaluation of phenotypic similarity studies and their impact on discontinuity systematics. Other than the first one, the criterion of reproductive viability, none of that's been done, as far as I know. And again, if somebody's got a paper that shows different, let me know. But as far as I know, nobody's touched any of this. None of it. It's all just not been touched at all. None of it. And that's really sad, because Walter ReMine, he wrote an excellent book, by the way. I'll get to that in a minute. I may do a review of it at some point. But ReMine is probably one of the greatest minds in biology creationists have produced. He was certainly one of the greatest minds in the field of systematics that creationists have produced. And yet, most of his work is undone, untouched. And that's unfortunate and sad. Because ReMine is just... These projects could be done. Like, some work has been done on the partial reproductive viability criterion. So some work's been done on that. But these projects could be done. There are people out there who have the skills to do them, but they are not doing them. They're doing, I hate to say this, but worthless statistical baraminology studies. Right? And it's unfortunate because statistical baraminology is, well, a raging dumpster fire. And we had a potential tool here. We had 2 forks in the road. We had statistical baraminology, which was Kurt Wise's project, or baraminology, which was Kurt Wise's project. It was made statistical by Robinson and Kavanaugh. And we had discontinuity systematics, and we picked the wrong fork. And it maybe discontinuity systematics is also the wrong fork. I don't know. But I know that the fork we've gone down for the last 30 years has been wrong. And that needs to be corrected. We need to take ReMine's work a little more seriously. Final paragraph. The accumulation and comparison of all this data will give scientists their first chance to see the living world through a systematic method that bears on the modern creation model. I encourage creationists to embrace this new biosystematics and begin the laborious task of resystematizing the life on Earth. All right, so that is ReMine's paper. This is a little bit of a long video. I recognize that. This is not a James Lindsay-worthy podcast, but it's not bad. Now, there is, at the end of this, I don't know if they still do this, but at the time, they did comments. There were comments at the end of the paper. I don't know if these are still included in the ICC. I'm not going to read the comments, But it it might be worth the time to read those comments if you're particularly interested in this and get an idea of the back and forth that went on between Wayne Frere, George Howe, and Kurt Wise. I'm fairly certain Frere and Howe are both dead, and now Kurt Wise and Walter ReMine. So I think it would be very interesting to see that discussed further. All right. So thanks everyone for tuning in. I'd like to see, just before I go, I'd like to see a lot more work done in discontinuity systematics, just to see if we could maybe fix some of the mistakes that we've made over the last 30 years. And hopefully my reading of this paper will jog somebody's brain to get the job done. Because I can't do all of this. I can do some of it, I can fight some of the rot, but I can't do it all. And I have other interests too that I need to pursue. Other things that need to be discussed, which is why we're going to be discussing Galuza and Gaskell's CET paper. But that's my encouragement for you all. Take some time. If you're equipped in this field, put some thought into it. If you have some ideas and you say, hey, I'm really interested, feel free to reach out to me. My email is available publicly. But if you don't have it, it's in his image. And that is two separate words, by the way. It's in his image at gmail.com. So feel free to reach out and discuss if you have some ideas on how to put ReMine's papers into practice, and we may be able to pull something together. All right, thanks all for tuning in. Hope to see you all next time.